Dr. Scott Fisher, speaking to you from Eugene, Oregon, where we're taking part in the University of Oregon Undergraduate Research Symposium. And we're happy and proud to be live streaming our poster presentation session today. And our session is named To the Moon and Back, Relativity Matters. And as an astronomy professor, of course, I like the title quite a bit. And I'm happy to say that we have 5 presentations from students that are studying physics and, among other things, here at UO. And we're going to do, like we've done before today, have a nice little poster session where our students have, a few minutes, 5 or 6 minutes to talk about a project that they've worked on. And then we're going to have a little Q&A at the end. I'd like to say that this is an informal session. We all know each other and study with each other. So it's nice to see some friendly faces on the screen. I'm being assisted by my colleague, Kathleen Freeman from the Computer Science department here at UO. And we're happy to talk all things physics here for the next hour or so. And everybody who's watching online, welcome and thanks for coming. Let's get right into it. I think that what we've gone over a few logistical matters. Each of our students is going to have control of the session while they're speaking, and we'll see. We're going to go into a full screen mode, and let me have a moment to introduce all of our speakers. Our first speaker is Noah. Noah has the title of his poster is called "Characterizing the Relationship Between Bacterial Motility and Range Expansion." As you can imagine, this is a project that spans the realm between physics and biophysics. And Noah, would you like to take it away for us and kick off the session?
Yes, I can do that.
Yeah, go ahead and share your screen. Thank you, sir.
All right, so you can see that?
We sure can. It looks real good on this end.
All right, cool. Well, thank you for introducing me. So today I'll be talking about characterizing the relationship between bacterial motility and range expansion, a project that I've been working on with my mentor, Dr. Raghu Parthasarathi. So a little bit of background. We decided to study cellular motility because it's a very well understood trait in many microorganisms, but in bacteria, its relation to larger scale range expansion hasn't been rigorously tested. So we decided to focus specifically on run and tumble motility, which is characterized by longer runs where a bacteria will travel kind of in a straight line, and it's then followed by short bouts of tumbling where the cell will randomly reorient itself, usually in response to a chemical gradient. So many current diffusion models that are used to explain how cells spread and move through a medium do not really incorporate the structure into their predictions. So we decided to focus on that as part of our study. So in order to perform this experiment, we decided to analyze 5 different bacterial species, and we measured range expansion in swim plates, which you can see in figure 2 there, right around here. I don't know if you can see my cursor. And we also used cellular tracking assays we examined cellular tracking assays using light sheet fluorescence microscopy. And here in figure 1, you can see an image of what that typically looks like. So to perform the analysis needed to make the measurements that we wanted, we performed particle tracking to measure speed and tumble time distributions, similar to what you would see in figure 3 here, from the data collected in cellular tracking assays, like in figure 1. And we also performed image segmentation on the swim plates, like in figure 2, to measure the range expansion rates and see how fast the bacterial colony expands on a plate. And that gave us graphs similar to figure 4 down here. And using both of those measurements, we compared using the diffusion coefficient and other quantities, how well matched they were. And initial results that we were getting suggest that they do not agree, and that current diffusion models are not enough to explain bacterial motility on a larger scale. So here in figure 3 here, we have a speed distribution for a particular species that we study called Aeromonas. And this graph suggests that the species has a mean speed of about 30 microns per second. And down here in figure 4, we can see a graph of the radius of a bacterial colony over time. So this shows the size essentially of the colony over time. And we can see it's a very linear relationship, although it's not perfect, and we can estimate what the expansion rate is. This particular graph gives us an expansion rate of 0.27 microns per second. And when compared, those two quantities do not agree with the predicted rate. So we don't have a ton of data yet, but in the future, we hope to increase the robustness of our tracking analysis by just improving the programs we've been using. And we also hope to improve the image segmentation to provide more accurate and improved measurements. We also hope to examine multi-species interactions and how different species of bacteria interact motile behavior. And we also want to measure the spatial diffusion within structured regions at a cellular level. So in figure 1 here, this is at the cellular level, and you can see in the images that we have collected that there seems to be some sort of structure there, and we hope to kind of measure how the bacteria move through that structure. So I would kind of like to thank Vikram Sandhu, Elena Wall, the Partha Sarathi Lab, and the Gilman Lab for all of their support in this research. You all have been a great help. So thank you.
Thank you very much. I think we're going to, as we're going to see 4 or 5 very interesting presentations, and each one comes at physics at a slightly different angle. And so I'll tell you what, let's hold our questions to the end, if that's okay. And this includes the folks who are watching online, if you can make a note. And we're going to have maybe 15 minutes or so at the end of the presentation for questions. So Noah, thank you very much, and hold tight. And I want to have at least one question for all of you. I've already seen some of your presentations, and so stick around to the very end. And thank you very much for that. Secondly, we have one on online. We'd like to go. One of our colleagues, Daniel, isn't here yet, so we're gonna, if it's okay, Laura, how are you feeling? Would you be ready to present for us right now?
Sure, I can go.
That sounds great. So let me introduce Laura, also from the physics department. And Laura is doing some high energy physics experiments, and the title of her presentation is Supersymmetric Long-Lived Particle Search using proton-proton collision data and simulations from the ATLAS experiment. And ATLAS is part of the Large Hadron Collider and CERN in Europe. Oh, and Noah, by the way, your time is up. Thank you very much for being a little bit early. And we will designate Laura, you get an extra 30 seconds. And so thank you very much. Laura, if you can share your screen and take it away, that would be wonderful.
Is that working all right? Great. So before I start, I just wanted to mention that I'm going to be doing more of a high-level overview and focusing on the background behind the research and then the final results, rather than digging into the details of the results as much, because I think that will help give a better idea of why it's important to do this research in the first place. So, bit of background. There are many unsolved problems in physics right now that make it clear that our current models are insufficient and there's a need for new physics. One of the models that could help with that is called supersymmetry, which basically says that there's a symmetry between the two fundamental kinds of particles, bosons and the fermions. And this symmetry implies the existence of a bunch of new particles, some of which have long lifetimes. Let me zoom into our background here so we can kind of follow along with the poster. The particular particle that is the focus of our search is called an R-hadron, which is a long-lived supersymmetric particle, and it consists of one supersymmetric particle and one or several standard model quarks or gluons. Those are the particles that we already know are the ones in the standard model. We've already found them. And R-hadrons decay to a neutralino, which is another supersymmetric particle. It's the partner of the neutrino, as well as some quarks. This neutralino passes through the detector without being noticed at all, and it carries with it a large amount of energy. And that means that events involving R-hadrons have a lot of missing energy in the final state. So when you reconstruct the event at the end, there's a lot of missing energy. And we quantify this with a variable called MET, which is specifically the missing energy in the transverse direction of the detector. Because our hadron events have this large MET, it's a really good sort of a key for trying to find them in searches. Another important bit of background that I want to talk about is some of the specifics of the ATLAS detector. ATLAS is a particle detector at the LHC, like Dr. Fisher said. It detects proton-proton collisions. And it has two main methods of reconstructing jets. And jets are regions of high particle density that originate from a single quark or a gluon. And they are defined solely by the algorithm that's used to reconstruct them. And like I mentioned, there are two algorithms that ATLAS uses, EM topojets and p-flow. EM topojets is the main algorithm that's been used so far in long-lived particle searches, and p-flow is sort of the new guy in these searches. So we don't know very well what sort of effects switching to p-flow will have on our searches and how efficient our searches are. So that's the main question of my research. I'll go down to the questions. That's the main question of my research, is looking at specifically how the MET changes when the jets are reconstructed using p-flow. Along the way, several other research questions came up, such as trying to determine what the relationship is between the detector geometry, the R hadron's decay radius, and the MET, as well as how to find what the JVT is for jets from R hadrons. And JVT is a number that jets are tagged with that gives the likelihood that they came from the primary vertex of the event. So it's a variable that we have access to at the end and could potentially use in our searches. In our analysis, we used two sets of simulated proton-proton collisions. Each set contained the production of two R hadrons, each with 2000 GeV and 10 nanosecond lifetime. And the two sets differ by the mass of the neutralinos in the final state. And that's important because the neutralinos going away and taking all that energy with them, that's a big part of the MET. So it's good to look at that when we're looking in the analysis. Each of those two sets contains 4 different samples, which were each reconstructed using different variations of EM topojets and p-flow. And the variations are in the JVT restrictions that are placed on the MET. And our analysis went through and compared these variables and calculated efficiencies, all to try to determine what the final result is of what the impact of a switch to p-flow will have on our search. And I don't really know how much time I have, but I'm just kind of gonna zip over these. We can-
You got about two minutes.
Okay,
Yeah.
I think I might zip over these a little bit. We can talk about them if we have more time or if there are questions, but these are the inclusive MET distribution plots. Oh, hey, that happened. The inclusive MET distribution plots and the efficiency of selecting different MET thresholds, as well as the results from those two side questions. But I think the most important thing to get to is the main conclusion, which is that the new reconstruction algorithm for jets, p-flow, will not lead to any significant deterioration of our sensitivity to our hadrons. So this is great news. It means that we're going to be able to use the vast amounts of data that ATLAS has detected so far and will continue to detect to start properly doing our long-lived particle searches with real data. Additionally, we also found from the JVT searches that we may be able to develop a new technique for identifying our hadrons based on the JVT of their jets. So that is where the future of our experiment lies, is looking deeper into ways to use our hadron jet JVT to try to identify them as an additional way to look for them. How am I doing on time?
You're doing real good. You hit it just about perfect.
Okay.
Yeah. Well, thank you for that. Do you feel that did you get across everything that you'd like right there in that time?
I feel like that got across the most important parts. Yeah.
No, excellent. Because look, we'll have the nice thing is we're right on schedule, and so we'll have a lot of time to stick around and chat afterwards too. So hang tight if you would. Thank you, Laura. Yeah.
I'll stop sharing them.
Yeah, stop sharing yours, please. Thank you. We'll get to the next one. So next we'd like to bring Maggie Thompson online. Hi, Maggie. And Maggie is continuing the trend of having our physics. Oh, by the way, look, you were perfectly on time. Thank you very much, Laura. And Maggie continues the trend of the domination of physics majors in this particular session. But Maggie's got a bit of a different slant on her project. This is a project that blends outreach and education and the development of STEM in an interesting way. And the title of Maggie's project is the SETI Scouts Project, developing scientifically literate young women through an astronomy destination camp at Pine Mountain Observatory. And Maggie, why don't you take it away? And you've got 6 minutes to go. I'll let you know.
Awesome. Let me just share my screen. All right, can people see the poster? It's a little bit zoomed in. Okay, cool, I see a thumbs up. All right, hello everyone. As Dr. Fisher said, my name is Maggie and I'm a physics student. And this poster mainly discusses how facilities like Pine Mountain Observatory can be an awesome hub for science outreach, as well as a great tool to get the public engaged with science through fully immersive programming. So Pine Mountain Observatory is owned and operated by the University of Oregon in the Deschutes National Forest and has two main goals. The first goal is focused on undergraduate research, as it's important to prepare students for both graduate school research and the professional world. And then Pine Mountain Observatory's second goal is to promote science outreach. And this is done through things like public nights, but also programming like the Girl Scouts Astronomy Destination Camp that I'm gonna be talking about in this poster. So the Destination Camp is a collaborative effort between the SETI Institute, Girl Scouts, Pine Mountain Observatory, and it's funded by a grant from NASA. It brings high school age girls interested in STEM careers to the observatory to help them develop their outdoor skills while also helping them to gain confidence and inspiration to continue their interest in STEM into the future. So this takes place over about a week where the girls are actually at the observatory and we host various activities. So the first main type of activity to help them develop their STEM knowledge is through discussion-based activities like learning about optics, constellations, data reduction, taking data, and also through more hands-on activities, which are my personal favorite, like instrument use. So for example, they had practice building Galileo telescopes, as well as operating the 14-inch plane wave telescope, which we use for research. And then the girls also work to develop their outdoor skills through hiking, camping, outdoor cooking every day, and they actually also learned about geocaching. And then lastly, we wanted to help them kind of develop a broader understanding of science community and science leadership. So we did this first through more cohort building between the girls by providing more fun activities to help the girls develop a community with each other. So that in the future, they could kind of have a group that they could fall back on for encouragement. And then we also wanted to get them more experience with the scientific method. And the main way that we did this was through having them keep actually a journal where they could record things that they were curious about, things they observed, and things they learned, which I think kind of really gets to the heart of the scientific process and encapsulates the spirit of science by thinking about a question, exploring that question, and then writing it down and talking with it to other scientists. And then lastly, we wanted to encourage them to continue their interest in STEM. So we had peer mentoring activities, mainly run by me, to give them kind of a positive female influence that they could ask questions to, kind of ask what it was like to be a female studying STEM. And then also throughout the week, they could ask UO staff about what it means to be a scientist and what that would look like for them in their future. So with this program, we kind of had short-term benefits and long-term benefits. So if you look down in figure 6 over here, if you can see my cursor, a lot of the Girl Scouts actually gained confidence in their new knowledge by the end of the week. So they became more involved in public nights by operating telescopes and talking about space and what they learned with the general public who visited the observatory during these public nights. And then also there were long-term benefits, like I mentioned. So for example, one of the girls from the 2019 program actually contacted us and asked for raw data from the Planewave telescope to ask if she could kind of play around with it, which really shows that she's still passionate about working with data months after the program had ended. And then additionally, one of the girls from the 2018 camp asked Dr. Scott Fisher, who's the observatory director, who's here today, to give a talk to her class about being a scientist and being an astronomer. So just kind of to wrap it up, in the future, we're hoping to get more quantitative data through surveys so that we can see if there's sort of a correlation between the way we conduct programming and changes in the way we conduct programming, and seeing if there's a correlation between that and then the girls' overall satisfaction with the program. Because right now we have a lot of kind of anecdotal evidence, but we really would like to get a few more survey data sets to get more quantitative data. So yes, thank you. And I will stop sharing.
Thank you, Maggie. This is let me just say right now, we're halfway through and this is the easiest session I've ever moderated. I haven't had to pull any of you off the screen yet for going over time. Thanks a lot. And so look, you know, because of that, we have a very we're in excellent time right now, and so we can all take a moment. It's going really well. And Sylvia, what do you think? Are you ready to go?
Yes, I'm ready.
Great. So this is Sylvia Mason, again, a physics major. And interestingly, we have another connection back to CERN. And the title of Sylvia's presentation is Visualizing Topo Cluster Algorithms for the Global Trigger. And this has to do with the instrumentation at CERN. So Sylvia, take it away for us, please.
All right, I'll share my screen. Can you guys see that? Okay, so hi, my name is Sylvia, as Dr. Fisher said, and I have been doing research in particle physics. So basically there is a standard model that explains the fundamental components of matter, but this model is incomplete, seeing as the matter described by it only makes up about 5% of the entire universe. So the Large Hadron Collider is a particle accelerator that collides bunches of protons in the hopes of discovering new particles or forces, so that we might begin to understand this other 95% of the universe. So this, for example, was how the Higgs boson was discovered in 2012 by looking at these proton-proton collisions. And the Large Hadron Collider, the LHC, will be upgraded starting in roughly the year 2026. And this upgrade will increase the number of particle collisions per second, so up to 200 collisions every 25 nanoseconds. And after this upgrade, the Atlas trigger system will need to reduce the amount of data that we collect by a factor of 40, because we're taking so much data, we can't possibly keep all of it. So this trigger system for this detector will need to select the interesting events that we want to keep within about 10 microseconds, so very fast. And our group is designing an algorithm for implementation in firmware in this trigger system for Atlas to help select these interesting events. And this is called the topo cluster algorithm. And basically it finds the most energetic cells in multiple layers of the detector and groups them together to reconstruct particle showers from these collisions. And what this algorithm does is it finds cells with transverse energy greater than 4 sigma, and these are called seeds. And the algorithm then loops around the neighboring cells in 3 dimensions. And cells with transverse energy greater than 2 sigma are added to that seeds cluster. And here, sigma is the electronics and pilot noise added in quadrature. So we should get these little like clusters of bright spots, like in these plots right here. And my research is focused on creating accurate 3D visualizations of these clusters. And this will help us understand the details of the performance of this clustering algorithm. So for example, this is what a total cluster from this algorithm would look like. As you can see, the central cell colored in yellow is the seed of the cluster, and then the surrounding cells in blue and green are the neighbors that pass that second energy threshold of 2 sigma. And you can also see that the cluster has cells in 4 different layers of the detector, as you can see on the z-axis. So that's basically what this algorithm does. It finds like the highest spots of energy from these collisions, and these are useful for reconstructing particle showers and looking for jets and missing transverse energy, like Laura talked about. And these can help us look for new particles. There's a problem with this algorithm though. It basically allows clusters to keep growing without stopping. So we need to implement splitting criteria to break up the larger clusters so that they better resemble the parton shower. So right here, a dip in energy would indicate that we need to split up one single cluster into 2 separate ones. So basically the next steps for my project are to visualize different splitting criteria to see what works the best and investigate how we might go about implementing that criteria in this algorithm. And by doing this, our algorithm will help the trigger system select interesting events in an accurate and efficient manner so that we may hopefully discover new particles or forces. And I would like to give a special thanks to the Presidential Undergraduate Research Scholars Program for funding my project this year. Thanks.
Well, thank you. Thank you very much. That's very interesting, and a good shout out to PERS. That's a great program. So look, let me just ask a very simple question. I don't see Daniel who at the moment. I think that he got held up or couldn't make it today. So because of that, we have lots of time, and so let's feel free. Hold on a second. As a matter of fact, let's open up the questions, and we have I'm going to be an official MC right here and take a question from YouTube. As a matter of fact, we have a friend from or a question from a person named June Stone, and actually it's an interesting question. Is that what's an example of a real world application of your research? And if there's not directly one, then can you maybe speak a couple words about the relevance of it? And look, Noah, you went first. If we can, let's rotate through the students real quick. Do you have a statement? Can you say something like that?
Yeah, I can speak to that a little bit. So in my lab, we do a lot of research with zebrafish and specifically the microbial communities that exist in the zebrafish gut. And so my research in studying microbial motility and how spatial structure is involved in influencing that motility can be useful for us to like in pursuing further experiments with zebrafish and understanding like how the microbes are moving around within the zebrafish gut and what like specific aspects of spatial structure might be influencing their motility and ultimately like the zebrafish's health, I guess.
No, thanks. Let's actually Laura, what do you think? Do you have did you have a direction for you? And of course, it's Sylvia, by the way, since you two feel free to tag team this answer in some sense. What do you think?
I think for my research, the biggest application is what it comes down to in discovering new particles, because as I mentioned at the beginning, there's a lot of new physics that we don't know yet, and if we discover new particles that might be key parts of that new physics, that would be That'd be just great.
Yeah.
That'd be a really good discovery to be able to make for physics as a whole.
Yeah, I think a comment we can make about particularly the work at Atlas is this is something that we would call fundamental research. This is truly, we're trying to learn about how do atoms work, and a very fundamental question like that. Sylvia, what would you say? Do you have a comment on that?
Yeah, so I totally agree with what you said. And like, if we think back to that discovery of the Higgs, like that totally explains like where the mass of all other particles comes from, like why things have mass, which is pretty big. So I think this idea of trying to discover like new particles or forces would just like explain like a lot about the super basic fundamental things that we don't necessarily always think about, but it would just explain a lot about our universe.
No, I appreciate that answer. I think that sometimes we don't even know how to ask the correct question. You ask a question, well, why do things weigh things?
Right.
Yeah, and it seems like a funny, why do you have mass? You know, and so this type of research that by the way, it's really proud to see undergrads working on. That these are very, very big questions but very fundamental questions. And Maggie, you might have a slightly different angle on this particular question. How do you see the relevance of the project and research you're doing?
For sure. I think there's kind of two benefits to figuring out how to best run these types of programs. I think the first one is that having programs specifically aimed around getting young women interested in STEM, I think really equals the playing field. I think some of my peers might agree in the observation that there are not very many young women invested specifically in physics careers. And so this type of program is meant to kind of encourage young women to feel confident enough to explore these questions that we've been talking about today. And then I think just in general, it's really useful to at least get more young people interested in science in general, because I think it's really important to have people at least interested in science so that we have a better, more well-informed community in general. So.
Yes, ma'am. I can't help but make a supporting statement and say that related to the project that Maggie is working on in particular is the idea of something we call science literacy. And can we get folks who maybe don't want to be scientists, but can we get them interested in science and give them a welcome into the world to learn at whatever the level that they want to. So I appreciate that question. Oh man, this is great. I'm not even going to have to answer any questions because YouTube is lit up right now for you all. And Jessica forwarded us another question from Addie Fecker. It was the name of the questioner. And how did you find your labs and topics? Love it, by the way. And was it always something you were interested in? And just for fun, we're going to go backwards this time. Maggie, go for it. You spoke last. Let's rotate it around just for fun.
All right. Thank you, Addie, for the question. So I kind of started out doing physics research and mainly about like galaxy evolution. But I kind of had a project that I worked on and I kind of wrapped up a portion of that project, and I realized that I didn't really find it super fulfilling. But what I did really like was volunteering at Pine Mountain Observatory and interacting with, yes, Dr. Fisher's background. But I did really like interacting with the public, talking to them about space, because I feel like space is very unifying, that a lot of people love space. And so I love getting people more engaged with more science-y topics related to that. So I decided to focus a little bit more on the outreach side of physics and physics education rather than doing awesome, like very sciencey hard research, which I also appreciate.
No, I think we all do that. Silvio, how did you get working with it, huh?
Well, when I was an incoming freshman, I was in a program run by the U of O called North Star, and we got to meet a whole bunch of different professors in physics, and we had lunch with Dr. Stephanie Majewski one day, and she was telling us about her research, and I was just blown away. I was like, this sounds incredible, like that's so crazy. And so I sent her an email in October of my freshman year, and I was like, I was really interested in your research, and I was wondering if you had any availability. I had never heard of like the LHC or CERN or anything like before
Yeah,
this.
so you were brand new. Yeah.
I was brand new. And
Yeah.
so I emailed her and I was like, I think what you do like sounds really cool. Like, can I get in on this? And she was like, yeah, of course. And so I started doing research my freshman year, and I've just been doing that ever since.
Good for you. So I think maybe one positive point we can take away is sometimes being proactive and being bold and going out and asking. Sometimes that's the way forward. Laura, what about you? What you think? What did you have? How'd you get into this? How'd you get involved?
It was sort of a similar story. I met Dr. Laura Janti at a meeting of the Society of Women in Physics. And I got a chance to talk to her about her research. And it's particle physics is something I've been interested in for a very long time. And she gave me the opportunity to begin working on a project.
So again, but you were the one who's, look, you went to the meeting, again, you took the time to go to the meeting to meet the faculty and to get to speak with them a little bit. So I think there's a common thread that's starting to weave is the be available, and go and check out and listen to talks and talk about topics even if you don't know anything about them. Yeah. Noah, how about you in the biophysics forum? Was it a similar path or did you do something different?
Yeah, so Sylvia and I actually share a very, very similar path. I was also in the North Star program
Oh, good.
in a different year, but yeah, we toured a lab, specifically my mentor's lab, Dr. Partha Sarathy's lab, and I kind of like had this inkling of, oh, I'm kind of interested in biology and physics. I came in as a physics major, but we toured his lab and he showed us all these really amazing videos of like vesicles and bacteria and zebrafish guts. And it was really fascinating to see. And I was like, I kind of want to make those videos too. And so I sent him an email and I said, hey, I'm very interested in joining your lab if you have any spots open. And so since then, I've kind of been getting into the research and I've been pursuing this project.
Wow, actually, I'm glad to hear it. I mean, I hope you see the common thread between the answers, that it's be engaged and be involved in what you're studying, and don't be too nervous about maybe approaching a professor or somebody that you might want to work with. Sylvia, we've got can you see the questions? Can you see the questions? And oh, okay, I'll tell you what, I won't read the questions. Sylvia has a question from someone we know quite well, Dr. Janti, another professor in the physics department. Would you like to tell the folks the question and maybe give an answer?
Yeah, so Professor Janti is asking, can you talk more about the how the 3D visualization you've developed will help understand the optimum splitting criteria? That's a good question. So basically this this visualization tool that I've developed is really good for like validation, I guess. So I guess it's nice for us to see kind of like in an obvious way, like if there's like a dip in energy between cells in the same layer or between the LAR and tile calorimeters, if there's like that huge dip in energy, then we should probably split those. So I would say it's mostly for getting to kind of validate and like make sure that what we're splitting and like how we're going to do that is going to work the best, if that makes sense.
Are there other ways that the data has been visualized or has been presented in the past? Was it 2D, for example? Is this a new thing, the 3D way?
Yeah, so we have a lot of 2D plots. Basically, we're good at making those. We know how to make them. But because topo clusters are inherently three-dimensional, it's better to use these 3D plots so that you can kind of see all of the information about the cluster.
Interesting. And then our co-collaborator Kathleen had a follow-up question, which is actually the question that I wrote down. Is I'm just curious now, are you using specific tools to make these? Are you what language are you writing them in, or what tools are you using? Can you give us just a little bit of a summary?
Yeah, so I use Python and C++ to write all my code.
Okay.
We also have a software package called Root that I use that has like specific classes of things that helps make these plots that we use.
Gotcha. So you can pull a skeleton of a plot. You know, there are templates or something out there that you can use, and then you customize them for your own purpose.
Yeah, so there are like classes of like specific histograms.
Gotcha.
So like for a 1D or a 2D or whatever, you can use those specific things to make these plots. But I will say that this trigger system, everything's going to be implemented in firmware, so it's not like it's going to use like Python code to like choose these interesting events. They're all going to be implemented on things called field programmable gate arrays, or FPGAs for short. So that's where those algorithms will live eventually.
And actually, I have an idea, but I'm kind of curious. Is that do you know the reason why it's going to be hardware implemented like that? I'm going to speculate. I'm going to guess. I don't mean to put you in the spot. My guess is, of course, is because of the speed, right? We need the code has to make decisions very microseconds, you were saying, and so it's easier if that stuff lives, you know, kind of on the hardware. But I'm very willing to be corrected there if that's not the reason.
Yeah. So these FPGAs, they're good because they're fast, basically, and I think that's the main reason why we're using them in this trigger system.
Gotcha.
Yeah.
No, that was a good one. You got 3 follow-ups. That was a triple whammy. Yeah. Well, look, I would like to ask one question of the other 3 folks. Again, we've got maybe about 15 minutes left. We can cut off a little early if we run out of stuff. Noah, I'm sort of interested. It's a very naive question, but how do you make the tracks? Are you using a video camera or a high-speed camera? I'm just curious about that. Can you say a couple words about the technology of making following the bacteria?
Yeah, so we use a pretty like standard microscopy camera.
Okay.
And it's not a high-speed camera or anything like that. I think it's just a very like a high-resolution camera.
Right. But this is literally a camera on the back of a microscope, that sort of thing. Yeah.
Yeah, like we have our whole microscope set up, and then we have the little like camera brick, I guess, and it's pretty high resolution. And from that, we take videos essentially of the bacteria moving around in the liquid. What I showed on my poster was a very small snippet of what we see. We actually get this like massive, or not massive, a fairly large image of the bacteria moving around. And my mentor wrote a few programs that actually like track the bacteria, because that's the difficult part. But that's actually one part of the project that we're trying to improve, because we're not entirely sure if we're getting perfectly accurate tracks. So yeah, and that program that he wrote kind of puts those like lines on them so we can visualize the tracks. But there's other analysis that
interesting. But this is, you know, kind of 30 frames a second or 60 frames a second, you know, kind of, you know, what it would be typical video speeds, not a million frames a second or something like that.
Yeah, it's roughly like 30 frames a second.
Okay, so yeah, so fairly standard camera technology mounted to specialized microscopy technology, that sort of thing.
Yeah, exactly.
Yeah, interesting. So Laura and Maggie, you guys can't get away. I wrote down something to you two, and I'm sorry. I hope it's the right question, but Laura, can you use the new methods you guys are developing to look at archived data from Atlas? I mean, is that a thing? You can go back and look at this vast database that's already collected and run the new codes or the algorithms through it? Is that a thing?
Yeah, we can, once we have our routines for finding our hadrons, we can use data that's been collected in the past, as well as data that's going to be collected when it powers back up again.
Yeah, gotcha. I was just thinking, it's like, it's interesting timing that while it's down and no new data is coming in, maybe you can go back and scour the database. By the way, which is a very commonly done in the astronomy world, where there again are big databases that exist, new algorithms come up, and you'll go back and scan old data for supernovas that went off or things like that. Oh, that's encouraging to hear that that data can be used after it's been obtained. Yeah. And Maggie, I can't resist. You and I have talked about this program a lot, and but I'm gonna put you on the spot a teeny bit, and that is, what is one way that you would change the program for next year? Look, assuming we're back to physical and everything is okay, like we ran it the first 2 years. Do you have an idea about that? Well, how would you change the Scout program?
I think I have a little bit of a bias in the way that I answer this question. But I would say if there was a way to have activities that, how do I say this? That were, so if there were more opportunities, so if there's another student like me who is really interested in like science education and outreach, more opportunities to like teach a little bit more, because I definitely assisted a lot, but there were some, like in some of the discussion and lecture activities, if students were able to run those as well. And then I think one thing that I definitely would love to see after the fact in the surveys, is the survey data that we got was very subjective on the part of the Girl Scout. And so I would love if we could find a way to get a more objective view of the language, or not the language, a more objective view of the knowledge that the girls were able to develop. Yeah,
I appreciate that. And by the way, for the folks watching, often when you deal with projects that are funded by grants, you do what is called external evaluation, where you have somebody come in and help to assess that sort of data. And we haven't done that yet, but we're thinking about that in the future. So look, we're not going to let you go yet. We've got another question that came in from YouTube, from Jeff Thompson, and he asked, can you recall an experience when you really felt like you were personally making a unique contribution to having an impact? And so, we've got 5 minutes total. So, so we don't have to ask everybody. Did any of the 5 of you have a moment that you really felt like, oh my gosh, I'm part of this and I did something big? Did that happen for you?
I can go.
You go, Mag.
Okay. So there was, at the very end of the camp, there was one time that one of the participants asked me if there were different things that she could look for in her Galileo telescope that night. And I had to like come up with a couple of things like right off the bat that I thought she would be able to see from her like smaller telescope. And I gave her a star cluster, and it's usually kind of hard to find. It's in the W, that is the Cassiopeia constellation. And she was able to find it, and that made me really proud.
So you truly contributed in that sense, for sure. Yeah. Well, look, thanks everybody. Oh, Noah, do you have something you'd like to contribute?
Yeah, I can also respond.
Oh, please do.
Yeah, so over the last summer, when I was kind of just getting started on this motility project, my PI, my mentor, was suggesting that we, like me and Vikram Sandhu, the other undergrad that was helping me at this project, that we rebuild one of the microscopes that we had in the lab. It was our tabletop microscope, and it was meant to just kind of be a minimal setup for a light sheet fluorescence microscope. And it wasn't working super well. And so we basically took it apart and put it all back together. And it was really just like the best feeling ever when we got it all put back together. And like after like 2 weeks of trying, and we could finally see like the little bacteria moving around, and we knew what we were seeing, and we were like, that's a bacterium. And
I did this thing. Yeah.
yeah, and it was just the best feeling.
I'm glad to hear that. I think that from an experience as an undergrad mentor, I think what we can see is that sometimes students' experiences are varied, but all of you are contributing quite significantly to the projects. Now, some of them are small projects. Maggie's project has 3 or 4 of us working on it. The Atlas collaboration has 2,000 or 3,000 people. And so our contributions may be bigger or smaller, but we're all part of it. And look, I'm going to get us wrapped up here. Is there any final comments anybody would like to say? Laura, what about you? You were quiet here the last few minutes. You good?
I could address the last question.
Yeah, please do.
I think I was thinking about it, trying to think of a moment in particular, and I think when I made my first histogram of the distribution for MET for just one reconstruction method, I looked at it and it made me so happy, because I thought, I've seen things like that in papers, and I just made one.
I made one. Yes, you're my first one. Look,
So every histogram from then on has always been like, yay, another one.
no, good for you. And again, maybe folks in our audience, it's okay to snicker a little bit if you think about us getting excited for making plots and histograms. But I promise you, the moment that the histogram appears on your screen is a very exciting thing. When you like, I did that, I made that thing. And so good for you, good for all the students. I'm really pleased to hear that you've had that experience. And by the way, folks, I can say, look, I know these students. They're very honest and open people. There's no stories here, and this has been a real interesting peek into the projects. Look, thank you all for taking part. It's a ton of work, and thank you for being flexible with this unusual virtual symposium that we're doing this year. Now, your only homework I'm going to give you and everybody who's watching is to please share the YouTube link far and wide. Give us a thumbs up so we can help thank the students. And let me just say that we're real proud of you, and congrats on your projects. And thank you very much, everybody. It's a great session.